Hello, everyone. My name is Anthony Tahayeri, and I'm a member of the Bertasoni Lab at Oregon Health and Science University. And I'm proud to present the ToothonaChip, a microfluidic organ system that will reduce the need for animal testing in the field of dentistry. Dental cavities and caries are the most common non-communicable disease. Nearly 90% of Western adults have or will experience a cavity or dental decay in their lifetime. In 2015, the World Health Organization estimated that there were 2.5 billion instances of caries worldwide. And in 2018, we saw an approximate amount of 160 million treatments for these cavities. Cavities don't seem to be going away, and the dental material market, which is centered on treating these issues, was valued at approximately 113 billion in 2018 and is projected to grow to 159 billion in 2024. Developing new treatments is costly and difficult. For researchers in the dental field, our current standards include effectiveness and low toxicity in animal models. But for most dental research done in animal models, data is only available when the experiment is finished and the animal is killed. Fortunately, dentistry and medical research are reducing the amount of animal tests done purely for toxicity purposes. One way researchers reduce animal testing is by testing toxicity with cells in a plastic dish. These experiments are still crude and do not accurately indicate low toxicity in animals. Regulatory bodies are also putting more pressure on researchers to reduce animal toxicity testing. The EPA wants a 30% reduction by 2025 and elimination by 2035. This means we need better lab testing to close the gap that a reduction in animal testing will leave behind. We are able to close the gap with our ToothonaChip system, the first and only device for R&D of oral healthcare products and dental materials, able to replicate the biomaterial to tooth-to-cell interface for clinically relevant and accurate results in real time. By mounting a part of actual tooth in the clear device, we create a window into distinct chambers, the outside of the tooth, seen here in red, where a full dental restoration can happen, and the green inside of the tooth, where any cells would be. It is also possible to do drug treatment on the inside chamber and monitor the effects on the outside chamber. All of this is possible while watching and recording the experiment 24/7 under a microscope. Our streamlined biological engineering platform makes designing and implementing complex tooth models easier and faster. Researchers are able to continuously visualize experiments, opening up the possibility to different styles of experiments found in other fields that have not been possible in dental research. And because it's a microdevice, less material is used per experiment. This allows the researcher to run bigger and better experiments for the same price. Where traditional bench experiments are quick and cheap, they lack clinical relevance, requiring the use of additional animal testing. And animal testing requires additional time for sample preparation after the experiment has been conducted, while also being much more expensive. For example, the cost of a medium-sized 30-day animal experiment ranges between $3,000 and $5,000. With the ToothonaChip, we can conduct similar size experiments at a 10x reduced cost without the additional sample preparation time. Our ToothonaChip combines the real-time observation capabilities of a bench experiment and mimics the animal environment without the rest of the animal handling requirements. We at the Bertasoni Lab are a group of scientists and engineers associated with OHSU and have a 5-year track record of working together to develop innovative solutions for various medical applications in our field of research. This includes a root canal regeneration therapy, a stackable bone graft for large wound repair, and an engineered bone tissue. These are among our published works, multiple of which have been found commercially viable, receiving commercialization grants and incubator funding from the NIH and Knight Cancer Institute, as well as having provisional patents. For the ToothonaChip, we've published research regarding the device and have received an initial $40,000 from grants and funding mechanisms at OHSU to continue development. With this money, we're conducting prototyping, additional biological testing, starting customer discovery, and researching our market. We want our device to be in the hands of researchers. We've established collaborations with multiple academic labs to contribute to the scientific literature as we continue to refine our design. Next, we will sell the device to researchers who will publish articles, build the body of knowledge around the device. Then we will target dental material companies for adoption. What we're asking for today is corporate interest in our device. We're looking to evaluate if there's interest or not as we continue development and create a scalable production pathway. We see a path forward through SBIR grant proposals and want to strengthen our claim with the backing of interested companies. I want to thank you all again for your time. Once again, this is the ToothonaChip, reducing the need for animal testing in the field of dentistry, and we would love to take your questions.